Criteria of genetic material. Before Griffith experiment, many scientists argued that DNA is genetic material, and some of the scientists were argued that RNA is genetic material, and some of the scientists were argued that proteins are responsible for inheritance. But after Griffith experiment, it was clear that DNA is major genetic material in organisms. So first, let us see the criteria of a genetic material. Any material, when it is called a genetic material, so that is, the material should follow these criteria, then only it can be considered as genetic material. A material can be a genetic material or non-genetic material. How can you say? Based on this criteria, we can say that if a genetic material, if any material is following all these criteria, then it is called as genetic material. If it is not following, then it is not considered as genetic material. The first one is, the genetic material should have its own replica. A genetic material should undergo replication process, so that one DNA molecule can produce another DNA molecule, so that DNA is genetic material. One RNA molecule can produce another RNA molecule, that is RNA replication, so that is also genetic material. But one protein cannot produce another protein, so that proteins are failed to follow this criteria, so that proteins are cannot be considered as genetic materials. So genetic material should have its own replica. And second one is, a genetic material should be stable. A genetic material should be stable. So stability is very important. Comparatively proteins, RNA is more stable. Comparatively RNA, DNA is more stable. So that DNA is major genetic material, RNA is primitive genetic material. So next, in the case of Griffith experiment, we can see that whenever the S strain is heat killed, so but DNA was not destroyed, but proteins and other cell organelles are destroyed. But whereas DNA is not destroyed in S strain of heat killed bacteria. S strain heat killed bacteria we have seen. So whenever S strain heat killed bacteria are completely died, after that DNA molecule was not spoiled due to heat treatment. So that is the stability. In the same case, the proteins and other components of the cell organelles were, the other case of proteins and other components of the cells were destroyed because of heat treatment. But DNA was not spoiled. This is important this is the reason why we can say that DNA molecule is stable. So that these are the two things is followed by DNA, but not proteins. In the first two cases, based on the first two cases only, we can say that proteins are not genetic materials, because proteins do not have replication process. One protein molecule cannot produce another protein molecule, whereas one DNA molecule can produce its own another DNA molecule by its own. But one protein molecule cannot produce another protein molecule without help of mRNA, without help of ribosomes. On the ribosomes, from the mRNA, protein is newly produced, but a protein is newly produced on the basis of mRNA on the surface of ribosome, but one protein cannot produce another protein. So this, if one material produces another material of its same, then it is considered as replication. In the case of DNA and in the case of RNA, both cases, DNA also, one DNA molecule can produce another DNA molecule, and one RNA molecule can produce another RNA molecule. So that in these two cases, we can say that replication is possible, but in the case of proteins, replication is not possible. And in the second, stability also proteins is less, because in the Griffith experiment, whenever the bacteria was heat treatment is given, then DNA molecule was not spoiled, but remaining all the components of cell was spoiled. Next, should undergo mutation. A genetic material should undergo mutation. A genetic material should follow Mendelian rules. A genetic material should follow Mendelian rules. So these are the totally criteria of genetic material. When any material is following all these things, then we can say that it is a genetic material. If any material, any substance or any molecule is following all these four laws or these four criteria are applicable for any one particular material, means that particular material can be considered as genetic material. This is criteria of genetic material. Which one is better genetic material, RNA or DNA? Let us solve this question. Which one is better genetic material, RNA or DNA? This is the question. Let us see the solution. So RNA is, one way it is better genetic material. It is better genetic material in terms of transcription. In terms of transcription, RNA is better genetic material. But in terms of stability, in terms of stability and other characters, DNA is better genetic material. RNA is better genetic material in the terms of transcription. How? For example, a DNA is present in the cell. In gene expression, a DNA should compulsory produces one mRNA, so that DNA depends on mRNA only for gene expression process. So compulsory DNA directly cannot code for a protein. DNA is having only genetic information, that's all. DNA has only genetic information which can be transferred through only mRNA. There is no other alternate for DNA. In order to express a gene or a protein must have to produce mRNA. So that the genetic information present in DNA must have to be copied into mRNA. So that then mRNA participates in translation process on the ribosome surface. On the ribosome surface, it produces finally protein. So here DNA contains genetic information for preparation of proteins. We know that already DNA contains genetic information for preparation of proteins. So that DNA is having genetic information, but directly it cannot send that information into proteins. So middle two steps are there, transcription and translation. So in that process of transcription, DNA sends its genetic material, or DNA transfers its genetic material to mRNA. DNA transfers its genetic material to mRNA, and mRNA transfers this genetic material to protein on the surface of ribosome. This is in this way, but in this way our mRNA, DNA must have to be dependent on mRNA. But for example, mRNA is the major genetic material in one cell. Directly it can code for a protein, but it is not depends on DNA. RNA does not depend on DNA for transcription process or translation process or protein synthesis or to express. RNA in regulation of RNA expressing, so RNA will not be depending on DNA, so that directly it can code for a proteins. RNA can directly codes for a protein, but DNA cannot code for a protein directly without mRNA. So that in order to express, DNA must have to be dependent on mRNA. So that in the transcription wise, RNA is a better genetic material compared to DNA. In transcription wise, RNA is a better genetic material compared to DNA. DNA is more stable than RNA. As we have discussed, in DNA and RNA, which one is better genetic material in stability wise? Stability wise, DNA only better genetic material, and DNA is more stable than that of RNA. DNA is more stable than that of RNA. How? The first one is 2'OH group, lacking of 2'OH group. 2'OH group is absent in DNA. 2'OH group is absent in DNA. 2'OH group is absent in DNA. So what is 2'OH group? In the second carbon in this place, now this is ribose nucleotide, this is base, and this is hydrogen, and this is hydrogen. If here OH is there, that is 2'OH group. If OH group is not there, so that this is another phosphate 3' end, and this is CH2, and this is phosphate 5' end, and this is hydrogen. See, now this is one nucleotide. In the nucleotide, this is base, and this is 3' end, this is 5' end, and in this this is second carbon. This is second carbon. At the second carbon, generally one OH is present, means that is RNA. If OH is not there, only hydrogen is there, means that is DNA. Presence of this OH group in second place is free OH group. This is because there are total 4 OH groups are present in ribose sugar, and 3 are replaced by phosphates, 2 phosphates and 1 base. One base replaces first OH group at first carbon, and another phosphate group is replaces another OH group at third carbon, so that it is 3', and one OH group is replaced by another phosphate at fifth carbon, so that it is called 5'. So in this way, totally 3 OH groups are replaced by 2 phosphates and 1 base. But in the case of second OH group, the second OH group is left free in RNA. Because of this free OH group, it is highly reactive. This is highly reactive OH group, so that it doesn't have its stability. So due to presence of this OH group at second place, so 2' OH group is absent in DNA. Due to presence of this OH group in RNA, it is less stable. But in the case of DNA, it is not present. It is only hydrogen is present here. So that because of absence of that O or 2' OH group, DNA is more stable. DNA is more stable due to absence of 2' OH group. And thymine is also called 5-methyl uracil. Thymine is also called 5-methyl uracil. Thymine is also called 5-methyl uracil. The 5-methyl uracil, and it is better nucleic. It is thymine is a better nitrogen base compared to uracil. Uracil and thymine, if you see, uracil is uracil is the less stable nitrogen base, whereas thymine is more stable nitrogen base. So due to presence of thymine and 2' OH group is absent in DNA, DNA is better or stable genetic material than RNA. Stable genetic material than RNA. Watson and Crick discovered double helical structure of DNA. Watson and Crick discovered double helical structure of DNA. Who discovered nucleotide for the first time? Frederick Mischer. Frederick Mischer discovered nucleic acid for the first time, and he has given the name nuclein. Frederick Mischer discovered the nucleic acid for the first time, and he has given the name nuclein. And the structure of DNA is discovered by Watson and Crick. And at the same time, Watson and Crick proposed that it is not escaped from our notice that specific pairing we have postulated, which immediately suggested the possible copying mechanism of genetic material. This is exactly the statement of Watson and Crick. While they have discovered the DNA double helical structure only, they discovered that how DNA replication takes place. Whenever they have discovered the DNA double helical structure, they discovered the double helical structure of DNA. Double helical structure of DNA on the basis of X-ray diffraction data of Wilkins and Franklin. X-ray diffraction data given by Wilkins and Franklin, they have discovered double helical structure of DNA, Watson and Crick. And Rosalind Franklin, so Rosalind Franklin is also there. And Rosalind Franklin, he has died later, so that they got a Nobel Prize. Watson and Crick, they got Nobel Prize for discovering double helical structure. The Watson and Crick, they discovered double helical structure on the basis of the X-ray diffraction data of DNA given by Wilkins and Franklin. And at the same time, whenever they discovered double helical structure, immediately they have discovered DNA replication. At the same time, they have given one statement that it is not escaped from our notice. We have postulated a specific pairing, and immediately it suggested that the possible copying mechanism of genetic material. Watson and Crick, they have given one statement that it is not escaped from our notice that specific pairing we have postulated, immediately it suggested a possible copying mechanism of genetic material. So that they have discovered this DNA replication mechanism in semi-conservation method. So what is semi-conservation method? Semi-conservation means semi-conservation method. In one DNA molecule, if you see, one DNA molecule, whenever this DNA molecule is separated, then a new DNA molecule is produced. This DNA molecule produces this DNA molecule is separate into two daughter DNAs, then in each strand produces a new copy. This is the white color one is parental DNA, this is parental DNA, and the blue color one is daughter DNA. new DNA strand, parental strand and new strand. So that two parental strands are protected in, two parental strands are protected in two different DNAs, two different daughter DNAs. This is one daughter DNA, this is one daughter DNA. So semi-conservation method means the two strands of parental DNA, two strands of parental DNA, this is two strands of parental DNA will be conserved in two different daughter DNA, two different daughter DNAs, two different daughter DNA molecules, two different daughter DNA molecules. So this is called semi-conservation method. This is called semi-conservation method. So let us see an experiment for semi-conservation method. Messerson and Stahl experiment. What is the aim of this experiment? To prove that DNA replication takes place in semi-conservation method. Semi-conservation method. DNA replication takes place in semi-conservation method. So semi-conservation method only, the proper method for DNA replication that is given by Messerson and Stahl. So DNA replication is takes place in only semi-conservation method. To prove this process, they done this experiment. That is, they have done this experiment with E. coli DNA. They have grown E. coli DNA in NH4Cl medium, ammonium chloride medium. In this ammonium chloride medium, the only source of nitrogen is N15 heavy isotope. N15 heavy isotope. When they grow one particular generation of DNA into this, so that one particular generation of DNA they have grown in N15 nitrogen isotope, then each and every nitrogen present in the DNA, they become into labeled with N15. So that this is N15 strand and this is N15 strand. They grown one E. coli DNA in NH4Cl medium for many generations. After this, the N15 gets incorporated into all the DNA, all the nitrogens of this DNA molecule. Now this DNA is considered as N15 DNA. N15 DNA, N15 labeled DNA means what? In this no normal nitrogen can be found. All the normal nitrogen atoms are replaced by N15 heavy isotope. Now this is called as N15 labeled heavy isotope labeled DNA. When E. coli DNA grown in NH4Cl medium for several generations. NH4Cl medium, in this, for the new formation or newly forming DNA molecules, N15 is only the source of nitrogen. So that in presence of nitrogen, N15 molecule only found in this place. In this DNA is N15 labeled DNA, meaning is all the nitrogen atoms present in this DNA are replaced by N15. So this is N15. So that this is heavy strand. So when this DNA is left for 20 minutes, after 20 minutes what happens? After 20 minutes, the two strands will be separated from each other. The two strands will be separated from each other, but they have shifted this for N14 medium. N14 medium. They have shifted this molecule into N14 medium, so that in the N14 medium, these N15 strands will be N15 strands will be undergo denaturation process. Both will be separated. After separating, then what happens in the N14 medium? The new DNA strand will form here. This is N14 strand, new DNA strand. N14 strand will form. So this is N15 strand, this is N15 strand, and this is N14 strand. This is N14 strand. This strand is called hybrid strand. This strand is called hybrid DNA. So now this is, how many hybrid DNAs are formed here? Two hybrid DNAs are formed, because one strand contains the parental DNA, both strands of parental DNA contains N15 isotope. But when this parental DNA is shifted to N15, the parental strands will not be converted. The newly formed DNA molecules, the newly formed DNA strands will be in the N14, but N15 remains as it is, even in the N14 medium. This is already prepared N15 DNA, parental DNA. See, listen, already prepared N15 DNA. This already prepared N15 DNA is kept in N14 medium for 20 minutes. Within the 20 minutes, DNA replication happens. After this before this DNA replication, what happens? These two DNA strands will be separated from each other like this. Then after that, the newly formed DNA molecules will get this N14, not N15, because the medium is N14. The source of nitrogen is N14. So that what happened? The newly formed DNA molecule will get N14 in its strand. So that already N15 two strands are separated from each other, and each N15 strand will form new N14 strand. So that a DNA molecule which contains one heavy strand and light strand, one heavy strand and one light strand is called what? Hybrid DNA, hybrid DNA. And this is, in this way, in first generation, after first 20 minutes, two hybrid DNA molecules are formed. Later 20 minutes, what happens? Let us see. Later 20 minutes, same N14 strand, later 20 minutes, next 20 minutes. The next 20 minutes, what happened? These two strands will be separated like this. This is N15 strand and this is N14 strand. These two are separated, and now these two are separated. So the medium is not yet changed, N14 medium only. In the N14 medium they have changed, so that the newly formed DNA is light DNA only will form. So this light strand only will form. This is, in this way, in every place, in everywhere light strand, because it is in the N14 medium, so that everywhere new strand will be light strand only formed. So that here after even for 20 minutes, even after 20 minutes, how many hybrid DNAs are formed? This is one hybrid DNA and this is one hybrid DNA. These two are light DNAs. How to find out light DNA and heavy DNA? To find out light DNA and heavy DNA, in cesium chloride, CSCl, cesium chloride, so it has to be, the DNA molecule has to be centrifugated. So when in the CSCl, cesium chloride, if the DNA is centrifugated, then we can find out heavy chain and light chain, heavy chain and light chain. This is Michelson and Stahl experiment. So this experiment we can say that one parent strand, one parent strand is having both N15 and N15, but in the next generation after 20 minutes, the daughter DNAs are having only one parent strand, one new strand. In the semi-conservation method means, the two strands of parental DNA will be conservated in two different daughter DNAs. That means each daughter DNA will have one old strand or one parent strand and one new strand. So this is semi-conservation method, and it was proved by Michelson and